Why not? So just to give you an orientation, my PhD has been about looking at the locomotion of the Perissodactyla. What are the Perissodactyla, I hear you say? Here's a smattering of some of the Perissodactyls that we do know of. These range from some of the, I'm going to use this one, by the way. Some of the small early horse ancestors, tapirs, my favorite animals in the universe. And of course, the Paleoloxodon-sized Indricotheres there. Had to get that one in, Steve, sorry. Okay, so the Rhinocerotidae are part of the Rhinocerotoidea, and these are some of the representatives of the Rhinocerotoidea. So we see a range of body plans within there, but all of these, I promise you, are rhinos one way or another. If we then take that down to the Rhinocerotids, this is what we get. So we are going in the incorrect direction. We have modern black rhinos, for example, woolly rhinos, which many of us would have known about, but we also have tusked rhinos. We have giant unicorn-like elasmotheres. We've got a range of different body plans there. And of the modern species, we have 5 to 6. It depends on who you believe, although one has recently gone extinct, so technically it's almost certainly going to be 5. And they currently have a tropical to subtropical distribution, but as is evidenced by things like Elasmotherium, Coelodonta, they used to have a much broader range going from the Holarctic all the way across down into the tropical Indochina and of course Africa. Careful. There we go. And Perissodactyls themselves have something called a mesaxonic manus. Now, what is a mesaxonic manus? This implies that the center of pressure passes through the central digit of the hand during locomotion. Now, for example, here we have a rhinocerotoid called Metamynodon, which appears to have something more like a hippo situation, which is a paraxonic. This is exhibited in artiodactyls. But what we're more interested in, and what my student who performed this project was interested in, is the mesaxonic manus of Rhinocerotids. These three are all Rhinocerotids, and you can see already, as a slight spoiler, that there is quite a bit of variation going on in the manus there. And what is driving these changes, these differences in the manus morphology? And is it a size effect exclusively? Is it phylogenetic? Is it ecological? As we can see from this brief, very sort of rough graphic, rhinos are different sizes. Like, no shit. But if we only think of size as affecting the locomotor apparatus, we would expect to see robusticity of the shape of the locomotor apparatus to increase with increasing body masses. If we find a complete scattergun approach, then maybe we are picking up signals of phylogeny or potentially ecology. And this is the question that my student was looking into by doing a morphometric assessment of the metacarpal, the third metacarpal of Rhinocerotids, and comparing this against body mass estimates and phylogeny and ecology. And here we can see some of the ranges of ecology. In fact, this one here on the left, this is Teleoceros. It has been considered as potentially a hippo-like animal existing in aquatic environments, also living out in the plains, eating grasses. Here we can see a no-horned rhino, an Acerothae here by the riverside, but also there is a two-horned one there hiding on the fringes of the forest, and two modern species, I can't see my pointer, two modern species, black rhino out in a desert here, and a Sumatran rhino in very dense forest. So there is a large ecological gradient going on there. So how do we investigate this potential changes in manus morphology? Well, my PhD and my students' project were involved CT scanning, laser scanning and photogram scanning the bones. We had 78 specimens across 22 species, ranging from approximately 30 million years ago to modern taxa. Here's an example of me scanning a knot rhino. Here's an example of some photographs done by Christophe Mallet at the museum in Paris, who kindly came onto the project because his project is also on rhinos. And quick recap of geometric morphometrics. I used landmark-based geometric morphometrics for this project, applied homologous, operationally or biologically homologous points to the bones, removed the effect of scale orientation and location in virtual space, align them on top of each other, and you can explore differences in morphology based on the corresponding points. I utilized linear measurements based on regression equations, which, as we learned earlier, is futile. But we did it at the time. We didn't know your awesome results. But the good thing is that we were using the same measurements across a range of metacarpals and looking within that group itself. So any actual specific body masses, we're not going to be publishing. We're not going to suggest not going to suggest that any particular rhino is that size. We just wanted to use it as a comparison. For ecologies, firstly we had trawled the literature for stable isotope recordings for carbon and oxygen. And here you can see if we have both high carbon and high oxygen values from tooth enamel, this is more indicative of a dry open environment, and in reverse, if low values both is more indicative of a forested and a wetter environment. I also looked at Ferranti indexes based on the molars back here. This is predominantly a technique used for examining equids, looking at the advance of grasslands and response of artiodactyls and perissodactyls to feeding on grasses. Not that many rhinos are what I would typically call hypsodont, but the technique of performing this analysis is essentially the same, and you can still see a gradation between deep forest taxa and grassland taxa. And finally, for the phylogeny, rhinoceros phylogeny, if anybody knows anything about it, is a large mess. Rather than worrying too much about a specific phylogeny, branch lengths, phylogenetic signaling, etc., we split the taxa that we did have data for into these groups. These are the species numbers that we have, not the specimen numbers. But here we have modern one-horned rhinos, modern two-horned rhinos. These are Dicerorhines. This includes Coelodonta and the modern Sumatran rhino. Teleoceratines, I will get back to in a minute. That's those rather hippo-like taxa. Where are we? Acerotherines, by their very nature, have no horns. And elasmotherines are quite variable. These include very early running rhinos, but also the large one-horned rhinoceros from the Pleistocene. So what did we do for results? Well, we looked at this in a multivariate analysis, and we went with a principal components analysis approach. These are all the taxa for along PC 1 and 2. And for the rest of the talk, I'm going to narrow this down to the means per species, because otherwise there's going to be too many points and it's going to get terribly confusing. So the pattern is in general the same. Here we have, here we have the rather troublesome teleoceratines. As you can see, there's an enormous amount of variation, pretty much equal to the variation exhibited by all the other rhinos. These are the early elasmotherines. This is Protocerotherium. This is essentially considered a running rhinoceros. And this circle encompasses modern rhinos. Up here we have the white rhino, black rhino, and the upside down triangles are the modern one-horned rhinoceroses. This is a bit of an easier graph to interpret. So here we have a long, thin third metacarpal, a slightly shorter, fatter third metacarpal, and here we have a incredibly stunted metacarpal. So from looking just at this graph, we can see that PC1 is essentially an axis of robusticity, which is great going back to our previous thought process that robusticity will increase as body mass increases. The predominant reason that these teleoceratids are down here is if we look at the proximal joint the fourth metacarpal, we can see that this joint facet in Teleoceratids is one facet. It does not split as it does in all these other rhinoceroses. This is also observed in very early rhinoceros, such as Hyraceus. It's also exhibited in tapirs, because I wanted to get a tapir shout out there, because I studied tapirs. But this is essentially what is causing that split along PC2 between the other quite short, robust taxa and the Teleoceratans. That's a messy slide, thinking about it. But regressing the robusticity axis, the PC1, against log body mass, hypsodonty index, carbon isotopes, and oxygen isotopes. Neither of the isotopes really gave us any reliable signal, so we pretty much discarded that as an ideal ecological metric. Fortunately, hypsodonty index did give us a nice positive result. So down here we have the sort of more gracile, longer metacarpals of the Protocerathes. Up here we have the Teleoceratids. Here we have the grazing white rhinoceros and Chiliotherium. Body mass, as it turns out, was a mess if you include Teleoceras, the genus Teleoceras. If you exclude the genus Teleoceras, you get an incredibly good correlation. So that leads me conveniently on to troublesome Teleoceras, as it should be called. Mostly driven by Teleoceras hicksi. This is a species from the east of the US. This animal seems to be very hippo-like in its appearance, with these quite flat top proximal joints to the metacarpal. This spreads out or it allows a larger surface area for the carpal complex to transmit forces through the manus. And when we look at the general bow plan of these taxa compared to, say, a narrow-horned rhinoceros, you can see here it is quite long in the trunk. And I'm not certain if anybody's actually done any biomechanical assessments on that in rhinoceroses, looking at the trunk and the head masses versus the morphology of the manus. But it's a very good place to start for somebody who wants to understand what on earth is going on with Teleoceratids, at least. And then here you can see just a brief, this is radius length over humerus length, metacarpal length over humerus length. Teleoceratids, again, they're coming out very separate to other taxa, and up here you have your white rhinos and your Pleistocene rhinos as well. As far as, I'll tell you what, I haven't got much time, so if you want me to talk about all of this, beautiful graphs, which I did last night, I will happily show you later. But to get on to the conclusions, in the Myosin, shape and size seems to be decoupled between taxa like the Acerathes, which are more running rhinoceroses, and the Teleoceratids, despite the fact that they are approximately the same mass. Teleoceras is an exceptionally divergent rhinoceros genus. It has been a pain in my and my students' behinds for the past year. So any further mechanical or comparative analyses on this taxon would be greatly appreciated. Open habitat taxa do seem to have more stout metacarpals, and this is completely reversed in Ecwids. You see a much more elongate situation going on in Ecwids until they become monodactyl. In fairness, they do then broaden their metacarpals. But it seems like these two groups of perissodactyls are doing rather different things once they get out into open environments. And finally, there does seem to be, as far as the hypsodonty index at least is concerned, there seems to be a relationship between feeding ecology and shape, as you can see here with a white rhinoceros and a Sumatran rhinoceros examples. With that, it leaves me to thank lots and lots of people for giving me lots and lots of specimens, and these two wonderful funding bodies for allowing me to go to the museums and fund all my research. There are unicorns around with us today. They're just big, they're fat, they occasionally bring us coffee. They're called rhinos. Thank you very much.